In this lecture, we will explore cutting-edge techniques in cell biology. Topics include multiplex immunofluorescence, super-resolution imaging, antibody characterization, co-localization analysis, and more. By the end, you'll understand the significance of these techniques in medical research and diagnostics. Let's get started. Multiplex immunofluorescence is a powerful technique that allows the detection of multiple proteins simultaneously within a single sample. This technique utilizes multiple fluorescently labeled antibodies, each targeting a specific protein of interest. By employing antibodies with distinct fluorescence spectra, the proteins can be visualized and their spatial distribution within the cellular context can be studied. Multiplex immunofluorescence is particularly advantageous in situations where studying the co-expression and co-localization of proteins is essential. This technique has found extensive applications in cancer research, molecular pathology, and immune cell profiling. Super-resolution imaging is a breakthrough technique that surpasses the diffraction limit of conventional light microscopy. It enables the visualization of cellular structures and molecular interactions with exceptional detail, at the level of nanometers. By utilizing specialized microscopy systems such as stimulated emission depletion, STED, microscopy, structured illumination microscopy, SIM, or single molecule localization microscopy, SMLM, an enhanced resolution beyond the previously thought limits can be achieved. Super-resolution imaging has revolutionized our understanding of cellular organization, intracellular dynamics, and protein-protein interactions. This technique has significant implications in the study of neurobiology, immunology, and developmental biology. Antibody characterization involves a comprehensive analysis of antibodies to ensure their specificity and suitability for experimental and clinical applications. It includes the evaluation of antibody binding affinity, selectivity, and validation through techniques such as immunoblotting, immunoprecipitation, and enzyme-linked immunosorbent assay, ELISA. Antibody characterization is vital to ensure reliable and reproducible results in various antibody-based assays, including immunofluorescence. This characterization helps in obtaining accurate data and avoids potential artifacts caused by nonspecific antibody bindings or cross-reactivity. Co-localization analysis is a technique used to determine whether two or more proteins coexist within the same cellular compartment. By utilizing multiplex immunofluorescence and image analysis software, the spatial relationship and degree of co-localization between proteins can be quantified. Co-localization analysis provides insights into protein interactions, cellular processes, and molecular pathways. This technique has widespread applications in studying protein complexes, cell signaling, and disease mechanisms. Immunofluorescence in 3D cell cultures enables the visualization and analysis of cellular structures and protein expression patterns within three-dimensional tissue-like environments. By combining 3D cell culture technologies with immunofluorescence labeling, researchers can mimic in vivo conditions and more accurately understand cellular behavior. This technique is particularly useful in drug discovery, tissue engineering, and regenerative medicine, as it better represents cellular responses and interactions within a physiologically relevant context. Antibody penetration and small molecule detection are critical factors to consider when performing immunofluorescence assays. Antibody penetration refers to the ability of antibodies to access their target proteins within the cellular structures. Small molecule detection involves the localization and quantification of small molecules, such as drugs or signaling molecules, using fluorophore-conjugated probes or antibodies. Understanding the permeability of antibodies and detecting small molecules accurately are essential for reliable and informative immunofluorescence experiments. Intracellular signaling visualization involves monitoring cellular signaling pathways in real time using fluorescent markers or genetically encoded fluorescent proteins. By tagging specific signaling molecules or their downstream effectors, the dynamic changes in signal transduction can be observed using techniques such as fluorescence resonance energy transfer, FRET, or biosensors. Intracellular signaling visualization provides insights into cellular communication, signal cascades, and regulatory mechanisms. This technique has been crucial in understanding diseases such as cancer, diabetes, and neurological disorders. Immunofluorescence has revolutionized clinical diagnostics by enabling the rapid and accurate detection of specific antigens or antibodies. This technique is extensively used for the diagnosis of infectious diseases, autoimmune disorders, and cancer biomarker profiling. Immunofluorescence in clinical diagnostics offers high sensitivity and specificity, allowing the identification and characterization of disease-related molecules within patient samples. Its applications include immunohistochemistry, flow cytometry, and fluorescent in situ hybridization, FISH. Time-lapse imaging involves capturing a series of images over defined time intervals to study dynamic cellular events. This technique enables the visualization of cellular processes such as cell division, migration, and intracellular transport in real time. Time-lapse imaging is commonly combined with fluorescence microscopy to track protein localization, organelle dynamics, and cellular responses to stimuli. It provides invaluable information about temporal changes in cellular behavior, helping to decipher complex cellular processes. High-throughput screening is a technique used to rapidly analyze a large number of samples or compounds for specific cellular responses or molecular interactions. By integrating automated imaging systems, liquid handling robots, and advanced data analysis software, researchers can screen libraries of compounds, genes, or siRNAs in a fast and efficient manner. High-throughput screening has revolutionized drug discovery, genetic screening, and functional genomics, leading to the identification of potential therapeutic targets and the understanding of gene function. Electron microscopy immunolabeling combines electron microscopy, EM, with immunofluorescence techniques to study the ultrastructural localization of specific proteins within cells or tissues. By labeling proteins with electron-dense markers, such as gold nanoparticles, their precise subcellular localization can be visualized at high resolution. Electron microscopy immunolabeling provides detailed insights into cellular morphology, cell-cell interactions, and organelle organization. It has been instrumental in elucidating the structural basis of various diseases and understanding intracellular transport mechanisms. Single-cell analysis involves the characterization and profiling of individual cells within a heterogeneous population. By employing advanced techniques such as flow cytometry, fluorescence-activated cell sorting, FACS, or single-cell sequencing, researchers can investigate cellular heterogeneity and identify rare cell subsets. Single-cell analysis has transformed our understanding of cellular development, immune responses, and disease progression. It holds immense potential for personalized medicine, regenerative therapy, and precision diagnostics. In conclusion, the advanced techniques discussed in this lecture have expanded our understanding of cellular processes, protein interactions, and disease mechanisms. These techniques provide researchers with valuable tools to explore cellular systems, leading to groundbreaking discoveries in molecular medicine, diagnostics, and therapeutics.